of the online tuition that we're offering for free. Let's just go through quickly what KayScience offers you. So you can click on any of these videos, and if you don't understand the topics or you've forgotten it, you can watch the videos, and then at the end, you've got question time, and then you've got gap fill exercises, and then you've got exam style questions at the end of every video, as you can see right here. And then the answers are shown directly afterwards. So you can click on any of these videos and you can go between them very easily. Then you've got the quizzes below, where you can test your understanding of what you saw in the video and then mark live. So if you write the incorrect answer, it will correct it for you. And then all of these quiz scores are tracked here. So you see what you can do and what you can't do. And that's for biology, chemistry, and physics. And this will, if you watch all of our videos and attempt all of these quizzes, guarantee you grade 8 or 9. That is if you get all of these questions right and watch all of our videos. So let's begin. Let's now do some revision on enzymes. So enzymes is a really important topic. So we're going to start off with the lock and key theory of enzymes. This here is an enzyme. This here is an enzyme. And the enzyme breaks down substrates into smaller products, or it makes larger products from smaller substrates. In this example, we're going to show the breakdown of a substrate into smaller products. So this here is a substrate. The substrate is going to bind the active site of the enzyme. So this here is the substrate, and this is going to bind the active site of the enzyme. As you can see, these substrate shape and the enzyme's active site shape are complementary and they are specific to one another. So what happens is the enzyme is bound by the substrate, and that happens in the active site. So the substrate binds the active site of the enzyme. And the enzyme is what we call the lock, and the substrate is what we call the key. This is why it's called the lock and key theory, because the substrate that binds the active site of the enzyme is specific to the enzyme, just like a lock and a key. So if we show you this example here, this substrate is not complementary to this active site's, so this enzyme's active site. So the substrate will not bind the enzyme because the substrate is not specific to the active site of the enzyme. So it will not bind. So the substrate must be specific to the active site of the enzyme. So let's go back to our example up here. It's got the enzyme, the substrate has bound the active site of the enzyme, which I'm going to show you now. The active site is bound by the substrate. So this here is the enzyme substrate complex. Why is it called the enzyme substrate complex? This is because the enzyme is bound by the substrate, the enzyme substrate complex. So this is the enzyme substrate complex when the enzyme has been bound by the active site. Sorry, when the enzyme's active site has been bound by the substrate. And in this case, the enzyme then going to break down the substrate into products. So the substrate is broken down into products. You can see that here. These are the products. So products, what word do you recognize from this? What is produced? So what is produced? This is the product. And in this case, the active site was bound by a larger substrate, which then broke down the larger substrate into smaller products. But this can also be the other way around. So let's now do question time. So the enzyme is the, press the answer in the chat, is the lock. An enzyme, is it a biological substrate? I forgot to say this, is a biological catalyst, or is it a biological reaction? So I left this out, so it's a catalyst. It speeds up the rate of reaction without being used up itself. Okay, and it's biological because it happens in biological organisms. So an enzyme is a biological catalyst. What does the substrate bind? 1, 2, or 3? So the substrate binds to the active site of the enzyme. So what we're now going to do is look at a substrate molecule. These are substrate molecules binding the active site, and they are now being put together to form a larger product. So this is now what enzymes can also do. They can also form larger products. So this here is the enzyme. This is the active site. These are the substrate molecules. And the substrate molecules will then bind the active site of the enzyme to form an enzyme substrate complex. The products, as you can see here, is larger than the substrates. So enzymes can also build larger molecules from smaller ones. So this is still a substrate molecule because it hasn't been added to the product here. So let's now go through question time one more time. So the substrate binds what? Put the number in the group chat. The substrate binds the active site of the enzyme. The substrate can be put the answer in the group chat. Both of the above. It can be broken down to smaller molecules, but also the substrate can bond together to form larger molecules. A broken down substrate is called a product. Right, so this here is the gap fill that you get at the end of every video. We're not going to do that now. You can pause it here if you're watching it, and these here are the answers. So you can pause here if you're watching this on catch up. But what I do want you to do is spend 1 minute answering question 1 to question 8. And if you complete questions 1 to 8, just do question 9. So you've got 1.5 minutes to answer those questions. Okay, let's go through the answers. And here are the answers. So question 9 was a 2-mark question in the exam where they ask you to refer to the lock and key theory to describe it. It's really important, the key words here. Substrates bind the active site of the enzyme. In an exam, you have to write active site of the enzyme. So if we go back and look at this exam question, with reference to the lock of key theory, describe how substrate is broken down. Substrate binds the active site of the enzyme. Active site is most likely a mark on the mark scheme, so you must write that. Depends on the mark scheme, forms an enzyme substrate complex, and the enzyme breaks down the substrate into products. So this here would be maybe 2-mark exam question, 3-mark exam question on foundation, potentially a 2-marker on higher, but the key words here are substrate binds active site and substrate is broken down into products. Those are the key points. So what we're now going to do is go on to the next bit, which is the enzymes of food. See how quick that and easy that was? So what we're going to look now are the enzymes of food. This here is a starch molecule. This here is a starch molecule. And it's represented by many glucose molecules bonded together. This is a glucose, 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 glucose. The glucose molecules are what we call monomers. Monomers. Mono meaning one, mer meaning unit. Glucose is the monomer. And the polymer is starch. And the starch molecule here is broken down into glucose molecules. So the product of starch being broken down is glucose. So this here is the starch, this is the starch, and these are the glucose molecules. These are the glucose molecules, and this is the product. So what breaks down the starch? As you can see here, you've got the enzyme amylase. Amylase binds the starch. So the amylase is the enzyme that breaks down. So amylase breaks down starch into glucose? It's amylase. That is an exam question. State the enzyme that breaks down starch into glucose, amylase. And you can see here that the amylase binds the active site, the starch binds the active site of the amylase. So the starch is the substrate, which binds the active site, which is in black here, and that gets broken down into the product glucose. So let's fill in the key gaps here. Pause the video if you're watching and catch up. I'm going to go for it now. The substrate is starch, and that binds the specific active site of the enzyme amylase. Amylase, which is the enzyme, breaks down starch into the product glucose. So what is the substrate here? The substrate is what gets broken down or put together to build a larger product or smaller products. It's getting broken down. The substrate here is starch. The enzyme is amylase. Amylase is the enzyme and the product is glucose. So this here is the answer. If you're watching to catch up, you can have a quick pause here. So let's look at the next food group, protein. This here is a protein, diagrammatical representation. Proteins are broken down into amino acids. So these here are the amino acids. So AA stands for amino acids. The amino acids are the monomers, and the polymer is starch. Polymer is many units. Poly, many, mer units. Polymer is protein. And this is the enzyme that will break down proteins. So this here is a protease. Protease breaks down proteins into amino acids. Amino acids. These are the products of protein breakdown, amino acids. Scientists are clever. ASE, if the word ends in ASE in a biology exam, that is an enzyme. Or look here, protease. Ase, an enzyme, like amylase, but this has protein in front of it. Protease breaks down proteins into amino acids. So scientists are very clever. So if you're asked which enzyme breaks down proteins, it's protease. So pause the video here, so you can practice using the keywords. And if you want to go over it, if you're watching a catch up, just pause it. Just look for the answers. The substrate here is protein. The substrate, protein, binds the specific active site of the enzyme protease. Protease. This here is protease, the enzyme. And the protease breaks down the protein into the product amino acids. So the substrate is protein, and the enzyme is protease, and the product is amino acids. You need to know this for your exam. This could easily be worth 2 marks out of 60 in your paper 1. It could even be worth more if you get a lot of it on food groups and enzymes. So these are the answers here. So we're now going to go through the last question time. So which enzyme breaks down proteins? 1, 2, or 3? Do it in the group chat. Well, that is going to be protease. Amylase breaks down 1, 2, or 3. Starch into glucose, number 3. The substrate of proteases are, so what is it that binds proteases? 1, 2, or 3? Protein. So we're now going to look at the last food group, which is lipids. Lipids, aka fats. This is the hardest one because it's got the most products, 2 different types of product. So this is the only reason why it's harder. It's actually, the beginning bit's not that difficult. So this here is a lipid. The lipid is bound by a lipase. So this here is the lipid, the substrate, which is aka fat, and this here is the enzyme, which is a lipase or lipase, depending on your pronunciation. Lip, as you can see here, L-I-P, lipid. Ase, A-S-E, like amylase, protease, lipase, it's an enzyme. So you've got lipase. Lipase breaks down the lipid into, so it's the lipase here that breaks down the lipid into products, which is glycerol and fatty acids. So the black rectangle here, from here, represents glycerol, and these are the fatty acids. So the lipase breaks down lipids into glycerol and fatty acids. So pause the video here if you're watching on catch up. So let's go through the answers. The substrate, lipid, binds the specific active site. So it binds the specific active site of the enzyme lipase. Lipase breaks down lipids into the products fatty acids and glycerol. Fatty acids and glycerol. Substrate is lipid, the enzyme is lipase, and the products are glycerol and fatty acids. So we've got one last question time here. 1, 2, or 3 in the group chat. And that is going to be lipids are broken down by lipase to fatty acids and glycerol. So products of the breakdown of lipids are, this one's quite easy, fatty acids and glycerol. So you can pause the video here at the end of each video. You've got gap fills, and these are going to be the answers. So pause it if you're watching on catch up, and these are the answers. Right, here we have, here we have some questions. I just want you to answer questions 1, 2, 3, 4, 5. Sorry, not question 5, not question 5. Answer 6, 7, 8, 9, not question 10, and not question 15, because that'd be silly. So I want you to answer questions 1 through to 4, and 6 through to 9, and then 11 through to 14. And you've got a minute to do that, a minute and a half. They're all one-word answers. So here are the answers. So I'll give you 10 seconds to go through the answers. And you can always return back to this later on. Okay, so let's now move on to a really fundamental part of enzymes. So what we're going to look at now is the denaturing of enzymes and what that means. So this here is an enzyme, and we're going to look at the word denaturing. Denaturing is this, so the active site of an enzyme changes shape. That's simply all at this level we need to understand, is when it changes shape. The more the enzyme's active site changes shape, the more it's denatured, so the less substrate combined. So substrate combined this enzyme's active site fully. So as you can see here, this substrate combined this active site fully. The substrate does not denature, okay? It is the active site of the enzyme that denatures. It's the active site of the enzyme that's denatured. And if it's only denatured a little bit, some can bind, some can't. There'll still be some breakdown of the substrate by the enzyme. It would be much slower, okay? And this is a fully denatured enzyme, and it will not bind the active site at all. So the enzyme here will break down this substrate at a faster rate than this one and this one. Okay, if you have a slightly denatured enzyme in your body, that means that that enzyme won't be as effective. And if your enzyme is fully denatured, it won't work at all, and it will not break down or make, it won't break down the substrate whatsoever. Okay, so denaturing. Okay, what part of the enzyme changes shape when it denatures? 1, 2, or 3? That's going to be the active site. The changing shape of the active site of an enzyme is called, this one's quite easy, denaturing. Denaturing when the active site changes shape. When active site denatures, what can no longer bind it? What can no longer bind the denatured active site? That's going to be the substrate. So as you can see here, you've got, you've got the gap fill that you can fill in, and then you've got questions here. We're not actually going to do these questions, apart from, I'm going to go through question 5 to you. So what happens to the volume of product formed when an enzyme denatures? So of course, let's go back to our diagram. So what happens to the product, the volume of the product formed if the enzyme denatures? This enzyme will break down this substrate more effectively and more quickly. So more product will be formed more quickly. So the volume of product will be higher when this enzyme breaks down this substrate compared to this one, which will produce less product over the same amount of time. So it'll produce a lower volume of product. Okay, so that there is denaturing, and the answers always follow on from the questions. We're now going to get into what I think is the most important bit. So today we're going to look at the effect of temperature on enzyme activity. All right, and this is so important because it underpins why it's a bad idea for humans to have fevers, because it can affect the way your body functions and the enzymes. And so let's have a look here. So what you've got is the rate of reaction on the y-axis, and you've got the temperature on the x-axis. So as you can see here, I am increasing the rate of reaction as you increase the temperature up until the optimum temperature. And then after the optimum temperature, the rate of reaction decreases. The rate of reaction decreases. And we're going to explain why this happens. We're going to explain why this happens. So what you've got here is the optimum temperature. At the optimum temperature, this is where the rate of reaction is at its highest. This is where the rate of reaction is at its highest. So the optimum temperature is not the highest temperature. It's the rate of reaction where it is, where the rate of, it's the temperature where the rate of reaction is at its highest. And in this case, it's going to be around 37 degrees. It's around 37.5 degrees. That's the optimum temperature, where the rate of reaction is at its highest. So that's so important. Now, kids that writes the highest temperature, it's the most frustrating thing. It's not the highest temperature. It's the temperature where the rate of reaction is at its highest. And in this case, it's 37.5 degrees. So let's now move on to explain what happens if you increase temperature beyond the optimum temperature. So as you can see here, as you increase the temperature from 37, which is the optimum, to 50, the rate of reaction decreases. The rate of reaction decreases. And at this point, the enzyme is fully formed. The active site is not denatured. But as you increase the temperature above the optimum, you can see here that the enzyme's active site begins to denature, which means that the substrate cannot bind the active site as well. But it's not denatured enough, so it won't bind it. As you increase the temperature above the optimum temperature more, the enzyme's active site denatures more and more until it gets to a point where the enzyme is fully denatured. As you can see here, the enzyme is fully denatured, which means that there is zero rate of reaction. So what we're going to do here, go through that one more time. So the optimum temperature is the temperature where the rate of reaction is at its highest. It's the optimum temperature. And as you increase the temperature above the optimum, the rate of reaction decreases because this is 1 mark in the exam, because the active site of the enzyme denatures. Therefore, the substrate no longer binds the active site. Therefore, there is less product being formed, which is an indication the rate of reaction decreases. So let's now do question time. Which part of the enzyme does the substrate bind? In the group chat, that's the active site. Right, the optimum temperature is what? It is the temperature with the highest rate. So increasing the temperature above the optimum temperature causes, now never confuse this one, remember the substrate does not denature. The rate of reaction decreases. Increasing the temperature above the optimum causes both of them. The active site of the enzyme denatures and less substrate binds the active site. Let's see what happens if the temperature decreases below the optimum temperature. So let's see what happens what happens to the rate of reaction if the temperature decreases below the optimum temperature. The rate of reaction decreases. You can see that here. But why? Why does the enzyme no longer break down the substrate as quickly? But the optimum temperature, you can see here that it's moving with a higher, it's got more energy stored in its kinetic energy store, so it's moving faster. And if you cool the enzyme down, let's say to this temperature here, you can see here that there is going to be less energy stored in the kinetic energy store of the enzyme and the substrate. So this is the enzyme and the substrate. So these speed lines represent the kinetic energy stored in the enzyme and the substrate. So there's less energy stored in the kinetic energy store of the enzyme and the substrate. This means that there are fewer collisions. Imagine you're in a classroom and there's 30 kids running around as fast as possible. You're going to collide with each other really quickly. But what if you just started to slow down and you walked around the classroom? There'll be less collisions between you and your friends. So if you decrease the temperature, that means that there's going to be less kinetic energy stored in the kinetic energy store of the enzyme and the substrate, which will lead to fewer collisions between the enzyme and the substrate, which leads to a lower rate of reaction and less product formed. So at this point here, 10 degrees, you're going to have even less energy stored in the kinetic energy store of the enzyme, which means that they're not moving as quickly, which means that there's less collisions between the substrate and the enzyme, which means that there is less product being formed, indicated by a lower rate of reaction. So if you decrease the temperature, you decrease the kinetic energy stored in the enzyme and the substrate, which leads to decreased kinetic energy of the enzyme and the substrate, and you've therefore got fewer collisions between the enzyme's active site and the substrate. Okay, so you need to know the difference between the two. If you go above the optimum temperature, not pH, so we'll look at pH in a second. If you go above the optimum pH, that means that the enzyme denatures, the active site changes shape, and then the substrate can no longer bind the denatured active site. If you go below the optimum temperature, it's about there being a decreased temperature, which leads to there being less kinetic energy stored in the enzyme and the substrate, therefore fewer collisions, therefore the rate of reaction decreases. Do not confuse the two. You're more likely to be asked, however, about the temperature going above the optimum temperature, but be prepared to answer this bit here. So let's look at question time. Oops, that was a bit too fast. Let's go back. So if the temperature decreases below the optimum, the enzyme doesn't denature. That's so vital. If the temperature decreases below the optimum, the kinetic energy of the enzyme and substrate decreases. If the temperature decreases below the optimum, less substrate binds the active site. And then the last question, I believe this will be, both of the above. So here's a gap fill, which I'm not going to do. And here are the questions. So you've got a few minutes to attempt these questions, and then I'm going to come back and go through them with you. Okay, let's now go through the answers. So these are all exam questions, by the way, all of them. Yeah. So here you go. These are the answers. And let's now go to the next video, which will be the last one today. So now we understand the effect of temperature on enzyme activity. This is going to be a lot easier, the effect of pH on enzyme activity. So what you've got here is you must always look at the x-axis to tell you what it is. This is the pH, this is the rate of reaction on the y-axis. So there's nothing about collisions and decreasing kinetic energy on this graph. This is all about denaturing. So you've got enzyme A here, and then you've got enzyme B there. You need to be able to identify, let's go back a bit, you need to be able to identify the optimum pHs. So the optimum temperature was the temperature where the rate of reaction was at its highest. And in this case, the optimum pH is where the pH, where the rate of reaction is at its highest. So this is going to be pH 2, and this one will be pH 8. We need to explain why the rate of reaction decreases above and below the optimum pH. And this one's much easier than temperature. So enzyme A and enzyme B, as I showed you before, you've got the optimum pHs of 2. So this is enzyme A is a optimum pH of 2. And this is the optimum pH of enzyme B. So we're now going to look at, I'm actually going to skip this one. Okay, let's now look here. So what happens if you increase or decrease the pH above or below the optimum pH, or the rate of reaction decreases either side? So the rate of reaction decreases either side. You can see that there. And it's the same on this graph here. The rate of reaction decreases either side of the optimum pH. But why? As you can see, this is an enzyme, and the enzyme is fully formed at its optimum pH. It's not denatured. You can see that it's not denatured. So it's not denatured. That's lock and key theory. I'm not going to go through that again because we've just done that. So we're going to skip on to this bit here. So let's look at this. This is the key bit. As you increase the pH above the optimum pH, it denatures. It denatures. And if you decrease the pH below the optimum pH, the enzyme also denatures. This is about denaturing only. So it's the same answer. Explain why the rate of reaction, it's an exam question, explain why the rate of reaction changes above the optimum pH, or something along those lines. Well, this is the answer. The enzyme, if you increase the pH above the optimum pH, the enzyme's active site denatures. You can see that there. The active site of the enzyme denatures. This means that, let's skip on to the next bit here. This means that less enzyme, sorry, less substrate, as you can see here, less substrate can bind the denatured enzyme. It will not bind it. Therefore, the substrate, which is here, is not broken down. We're going to skip the question time. It's the same, exactly the same on this one. The enzyme's active site denatures. It denatures as you increase the pH above its optimum pH, or if you decrease the pH below its optimum pH. And this means that the enzyme's active site can no longer be bound by the substrate. Okay, so we're now going to go to the end bit. I'm going to go through the questions with you. So, which part of the enzyme does the substrate bind? That is the active site. What is denaturing? It is the changing shape of the enzyme. It's the active site. It's the changing shape of the enzyme's active site. To find optimum pH, it is the pH where the rate of reaction is at its highest. What happens to the enzyme's active site if you increase the pH above the optimum? It denatures. What happens to the enzyme's active site if you decrease the pH below the optimum pH? Denatures. What can no longer bind a denatured active site? Substrate. What happens to the volume of product produced if the active site is denatured? Well, the volume of product decreases. So what this is here, question 8 is what I want you to do now, and it's an exam question. Explain why enzyme activity decreases if the pH decreases below the optimum. You've got 2 minutes to answer that question, and I'll go through the answers with you. Right, let's go for the answers. So explain why the enzyme activity decreases if the pH decreases above the optimum pH. Well, the enzyme's active site denatures, which means the enzyme's active site changes shape. So less substrate binds the active site, so less product is produced. And that there would be about 3 marks in an exam. So what I'm going to do now is just, so that's the end of the tuition. So remember, you can use the code here, K50, 50% off KayScience, any KayScience account. And that gives you access to our revisions. You can do formula practice. You can do quiz questions, so you can do like that. If you've got a Unit 1 topic coming up, you can choose 5 questions from each, and you can start the quiz. And then you can just answer the quiz there. You can finish the revision. You've got your formula practice. So go to formula practice, you've got that. You've also got converting units. So if you click on that, you can do that. And then remember, you can always track what quizzes you've done. And you can go between biology, chemistry, and physics. So 50% off, you can use the code K50, which is seen in the bottom left. Really hope you enjoyed that. Come back on Thursday for some more chemistry revision. So yeah, please like and subscribe as well. It's so important that you guys do that, and just tell your friends about us. So yeah, have a really nice evening. Thank you for watching. Goodbye.